Good morning. In Chapter 6, we will look at microbial nutrition and growth. This is a picture of my dog Bella when she was just a little itty-bitty baby. We'll see what happens to her over time. The question for this chapter is oxygen in the atmosphere is essential for human life. How then can some bacteria grow without oxygen being present? We'll answer that further in the lecture. When we talk about microbial growth, we are talking about an increase in the number of cells, not the cell size. Microbial growth is expressed as populations, which is the actual number of bacteria within a group, and colonies, which are groups of cells large enough to be seen on media without the aid of a microscope. For example, these are colonies. Bacterial colonies can take on different shapes. They can have different margins on the edges. They can be flat to elevated. I love the umbonate one. It looks like it has a little nipple in the middle of the colony. They can be different sizes, rough, smooth. Appearance can be glistening or dull. They can be pigmented or non-pigmented, such as this one that is pigmented red. And they can be opaque, translucent, or even transparent. Sometimes it is difficult to see bacterial colonies on the surface of the media unless you hold it just right in the light because the colonies are actually transparent. Chemical requirements for growth of any organisms, but especially bacteria, would include carbon, nitrogen, sulfur, phosphorus, certain trace elements, oxygen, at least for some, and usually some sort of organic growth factors. Physical requirements include the correct temperature, the correct pH, and the correct osmotic, and I'll put in here as well, hydrostatic pressure. Carbon plays many roles. It is found in all structural organic molecules, including carbohydrates, fats, proteins, and nucleic acids, and for many bacteria, it is an energy source. Chemoheterotrophs use organic carbon sources such as glucose, while chemoautotrophs use CO2 as their carbon source. Here's figure 6.1 on page 165 showing the phototrophs and the chemotrophs. And you can see the chemoautotrophs use carbon dioxide as their carbon source, while chemoheterotrophs use the organic compounds. Nitrogen is also needed in bacterial metabolism. It's used in amino acids and protein structures. Most bacteria can actually break down proteins and so release the nitrogen into the atmosphere. Some bacteria use NH4+, which is the ammonium ion, or NO3-, which is the nitrate ion. A few bacteria can actually use N2 nitrogen gas in a process known as nitrogen fixation. Sulfur is also found in amino acids, but found in some of our cofactors and coenzymes, thiamine and biotin, a couple of the B vitamins. Again, since most bacteria can decompose proteins, those that contain sulfur-containing amino acids will release sulfur. Some bacteria can use SO4 or hydrogen sulfide. Phosphorus, where do we find phosphorus? Well, it's found in DNA, RNA, ATP, adenosine triphosphate, and phospholipid membranes. PO4-3 is a source of phosphorus in most cells. Hydrogen is essential for hydrogen bonding and in the electron transport chain. Organotrophs are bacteria that acquire hydrogen ion from the same organic molecules that provided the carbon, such as glucose. And lithotrophs acquire hydrogen from inorganic molecules such as hydrogen gas, nitrite ion, hydrogen sulfide, and forms of iron compounds. Trace elements are inorganic elements that are required in extremely small amounts, such as iron, copper, molybdenum, and zinc. Most of these trace elements will actually be used as enzyme cofactors in the different enzymatic pathways. Organic compounds that must be obtained from the environment would include certain vitamins, especially the B vitamins, certain amino acids, purines, and pyrimidines to make up DNA and RNA. This table lists some of the growth factors of microorganisms. such as amino acids, components of proteins, cholesterol used by mycoplasma bacteria for cell membranes because they need more cholesterol in the phospholipid membrane because they do not have a cell wall. NADH is the electron carrier. Niacin, pantothenic acid, para-aminobenzoic acid, riboflavin, and thiamine. You'll notice that B vitamins play a major role here. The effects of oxygen on growth are varied depending upon what type of metabolism the bacteria does. Obligate aerobes require oxygen in the environment. If we put these bacteria into a deep media tube, they will grow best at the top of the tube where most of the oxygen is located. Versus facultative anaerobes that can produce energy through aerobic respiration if oxygen is present, but then switch to anaerobic respiration if the oxygen is not present and for minimal material is present in the environment. Typically, facultative anaerobes grow throughout the tube, but the best growth will be at the top of the tube. They prefer to do aerobic respiration if oxygen is present. You go back, remember aerobic respiration gives us more ATP in general than does anaerobic respiration or fermentation. So here we have the obligate aerobes growing at the very top of the tube where most of the oxygen would diffuse into the media. The facultative anaerobes, you do see growth throughout the tube, but the best growth will be at the top because these prefer oxygen, but you'll have some growth at the bottom where there's little oxygen simply because they can switch. Obligate anaerobes can live only in the absence of oxygen or live in low oxygen environments. They typically grow only at the bottom of the tube where there is much less oxygen present. Aerotolerant anaerobes grow under both aerobic and anaerobic conditions, but they do not shift from one mode of metabolism to another as conditions change. These are strictly fermenting bacteria, so they don't care if oxygen is present or not. Aerotolerant anaerobes will grow equally throughout the tube because it just doesn't matter the amount of oxygen. Then we have a group of bacteria known as the microaerophiles. These require oxygen at lower than atmospheric concentration, but not 0% oxygen. So they tend to grow best about the middle of the tube. Here again is the picture. So our obligate anaerobes growing right down here at the bottom of the tube. The aerotolerant anaerobes, again, don't care if oxygen is present or not, so they grow equally throughout the tube. And the microaerophiles here in the middle, where there's not as much oxygen as at the top, but more oxygen than at the bottom. Can you give a patient too much oxygen? And the answer is yes. What happens when oxygen becomes toxic? Singlet oxygen is the state where O2 is boosted to a higher energy state. If you go back to the atomic structure of molecular oxygen, you will see that both second orbital shells share 8 electrons around each nucleus. Singlet oxygen, one of those electrons gets boosted to the third orbital shell. And now he's out there by himself, just zipping around, and this is what we call the higher energy state. When you have this situation of this lone electron out there, this singlet oxygen then can actually tear through structures such as proteins, nucleic acids, and so this can cause damage within the cell. Superoxide free radicals is basically the same thing, except this time, instead of throwing one of the electrons out of the second shell, we simply have gained an electron from someone. So it's O2 negative. If you put 2 of these superoxide-free radicals in a cell that has the enzyme superoxidase dismutase, it will form H2O2, which is hydrogen peroxide, and oxygen. The peroxide anion has actually gained 2 electrons in the third shell. If we take hydrogen peroxide in the presence of the enzyme catalase, It will break down into oxygen and water. This is what happens when you put hydrogen peroxide on an open wound. The damaged cells have released their enzyme catalase, and this produces H2O and oxygen. The fizzing property is from the production of oxygen. If you take hydrogen peroxide with oxygen and put it in the presence of peroxidase, which is what this should say, you then will just simply get water. The hydroxyl radical, which is OH with a dot, normally we see OH negative, it has lost an electron. Even though this is an ion group normally, this is an uncharged ion. If anyone can explain free radicals to me any better than that, please do so. All we know is that free radicals, such as the hydroxyl radical, can cause tissue damage in cells. The physical requirements for growth include temperature. The minimum growth temperature is the lowest temperature at which the species will grow. The optimum growth temperature is the temperature at which the species likes to grow best, and the maximum temperature is the highest temperature at which the species will still grow. The effects of temperature on proteins will be denaturing. Both hot temperatures and cold temperatures will simply break down the structure of the proteins. Oh yeah, remember, all enzymes are structurally proteins. Also, temperature can have an effect on the lipid-containing membranes of cells and organelles. If the temperature is too low, the membranes become rigid and fragile and tend to shatter. If the temperature is too high, the membranes become too fluid. You can try this. Put some olive oil in a pan, set it on the stove. What happens? The oil begins to actually flow. It becomes more liquefied. This shows a graph of the temperatures at which this bacteria tends to grow. The optimum temperature is the best growth rate. The maximum temperature is where we still would have some growth, not good growth necessarily, but some growth, and the minimum temperature is the same thing. You'll notice that this graph is somewhat skewed. Higher temperatures tend to break down proteins and lipids much more quickly than does cold temperatures. In this series of plates at 22 degrees Celsius, 30 degrees Celsius, and 37 degrees Celsius body temperature, which has the best growth rate? Equal amounts of bacteria were applied to each of the plates. We can use temperature to then separate bacteria into groups. The psychrophiles are cold-loving microbes, having an optimal temperature for growth at about 15 degrees Celsius or slightly lower. The psychotrophs are cold-tolerant bacteria or archaea. They tend to grow at low temperatures but have an optimal maximal growth temperature above 15 and 20 degrees respectively for the bacteria and the archaea. These are perfectly happy in the refrigerator. The mesophiles are the moderate temperature-loving microbes. These include most of the pathogens because their optimum growth temperature is around 37 degrees Celsius body temperature. Thermophiles are heat-loving microbes. Many of these are what we refer to as hyperthermophiles, that are extreme thermophiles living in the hot springs or thermal vents associated with volcanic activity. Most of these tend to grow within a limited range of temperatures, with the minimum and maximum growth temperature only about 30 degrees Celsius apart. So, for example, the psychrophiles, maximum growth temperature around 12 to 15 degrees Celsius here, the maximum temperature about 18, the minimum temperature about -8. That's about a 30-degree span. The mesophiles, however, have a little wider base, about 40 degrees Celsius, anywhere from about 10 as the minimum temperature to about 48 degrees. The thermophiles run from about 40 to 70 degrees, and the hyperthermophiles are much greater than 60 degrees Celsius and go even higher than 110 that we now understand. Which one of these groups then would contain those pathogens for us? This is an example of a psychrophile. This is actually a sheet of Antarctic ice during the very balmy Antarctic summer. During the warmer period, these bacteria reproduce very, very rapidly, and they are pigmented red. They actually turn the sheet ice red. However, they can withstand the very cold temperatures of an Antarctic winter and do just fine. Psychotrophs grow between about 0 degrees Celsius and about 30 degrees Celsius. Most refrigerator temperatures are about 4 to 5 degrees Celsius. These are the ones that can cause food spoilage because they grow well in the refrigerator. This is an example of some of these bacterial contaminations in this tomato. I like this picture because it shows, both on the Fahrenheit scale and the centigrade scale, the danger zone for pathogens. You'll notice that from about 20 to 50 degrees Celsius, or about 60 to 130 degrees Fahrenheit, we have rapid growth of bacteria. And during this growth, many of them produce toxins that actually cause disease. But when we heat it to over 60 degrees, most of the mesophiles cannot survive. And so what temperature do you want to cook your meat at? Ground meat is to be cooked to 160 to 165 degrees. Why? Many bacteria can survive lower temperatures, even of the mesophiles, and may grow slowly. So in the refrigerator, some of them can actually still survive, but very few of them actually are pathogenic. And freezing, there's no significant growth of these mesophiles below freezing. Here's an example of a thermophile. This is one of the Bacillus species. It was isolated out of a compost pile that actually read 55 degrees Celsius. What category would this be? pH also plays a role in growth. Most pathogenic bacteria like to grow between the pH of 6.5 and 7.5. Why? Molten yeast grow a little bit more on the acidic side, about 5 to 6. Acidophiles can grow in acidic environments, anything lower than pH 7. Neutrophiles like to grow at around pH of 7. And alkaphiles tend to grow in basic environments greater than 7. So pathogens for us would be in which category, do you think? When bacteria are cultured in the laboratory, such as when I use your stock cultures of bacteria, they often produce acids that eventually interfere with the colony's growth within the broth media. They produce so much acids that they lower the pH of the media to a point that the bacteria can no longer grow. Pressure also plays a role. Osmotic pressures can play a role because of their damage on the cell membrane. Hypertonic environments or an increase in salt or sugar can cause plasmolysis. Why is it that our ancestors then tended to have salted or sugar-cured meats? Because this actually stopped bacterial growth. Obligate halophiles require very high osmotic pressures. Facultative halophiles can tolerate high osmotic pressures for a period of time, but typically tend not to like it as much. Plasmolysis, if you remember, occurs if the concentration of solutes, such as sodium chloride, is higher in the surrounding medium, causing a hypertonic solution, and water tends to leave the cell. The cell wall does not shrink, but the cell membrane will shrink and oftentimes will cause tearing of the membrane and leakage of cell content. Hydrostatic pressure. We know that water exerts pressure in proportion to its depth. For every additional 10 meters of depth, water pressure increases by 1 atmosphere. So what is the pressure at 7 miles down in the Marianas Trench, where these bacteria have been found in these volcanic vents? The barophiles require high hydrostatic pressures, such as found in these deep ocean basins and trenches. Associations in biofilms allow microorganisms to live in association with different species. We have different types of associations: antagonistic relationships, where the two organisms will fight each other; synergistic relationships, where the old saying that the whole is greater than the sum of its parts, they actually assist each other very well; and symbiotic relationships where both of them get something from the relationship. Biofilms are microbial communities that reside in a polysaccharide DNA protein matrix that forms a slime or hydrogel. Bacteria are attracted by chemicals via something known as quorum sensing that allow bacteria and other microorganisms to coordinate activity and group together to provide benefits for the individuals. So here's an example. We have some sort of organic layer attachment onto a surface. Let's say your tooth. This allows primary colonization of bacteria and attachment. This bacteria then can grow and produce more of the matrix, and that produces more attractants for other organisms. This is the way tooth plaque actually builds up. Here's an example of rock snot. This is actually a biofilm made up of bacteria and fungus and algae found on the surface of the rock. Biofilms can share nutrients among the different bacteria. They can shelter each other from harmful factors. If you're in the middle of this big glob of microorganisms, you're a lot less likely to be infected by the environment as the ones on the outer surface. And they can work cooperatively to carry out complex tasks for the community. One microorganism may produce something that another microorganism can use or need in its metabolism, and then microorganisms may produce something that the other guy can actually use. This is actually a scanning electron micrograph of some plaque on the human tooth. This is why you should brush. 70% of all human bacterial infections tend to involve biofilm formation. Most nosocomial infections, those acquired in a hospital or clinic setting, are probably related to biofilms that form on indwelling devices, such as IV catheters, urinary catheters, or other indwelling medical devices, such as artificial heart valves, artificial joint replacements, etc. This next slide shows an intraoperative photograph taken during excisional surgery to remove an infected prosthetic knee joint and shows a bacterial biofilm of an infected arthroplasty. The white material that the large white arrow is pointing to is pus and contains huge numbers of microorganisms and host-derived leukocytes, especially neutrophils. This is the number one reason why artificial joints have to be replaced from time to time, is the formation of biofilms on the artificial joint parts. Culture media provides nutrients prepared for microbial growth. And if you know the metabolism of your bacteria you're putting in there, you can put in the culture whatever the bacteria may need to grow well. The inoculum is the microbes introduced into a media that initiates the growth. So the small plastic tubes that I give you with bacteria are called inoculum tubes because they contain the inoculum. The culture is the microbes growing in or on a culture medium. If something is sterile, it means that there are no living microbes in or on the media. Agar is a complex polysaccharide that's long been used as a thickener in foods such as jellies and ice cream. Agar is basically gelatin. It's used as a solidifying agent for culture medias in petri dishes, the slant tubes, and the deep media tubes. Generally, because it is a complex polysaccharide, most microbes cannot metabolize the agar. Therefore, it is an inert substance in the media. It liquefies around 100 degrees Celsius and solidifies at about 40 degrees Celsius. When we sterilize the agar for the petri dishes, it comes out of the autoclave at 121 degrees Celsius. We then pour the liquid into the plates upside down and allow it to solidify to room temperature. It begins to solidify at about 40 degrees. By the time it reaches room temperature of 25 degrees Celsius, it will be solid and we can turn the plates with the agar up. It will not liquefy in the incubator at 37 degrees because it solidifies at a much higher temperature. Here's a good example of a very flavorful agar. Someone had a lot of time on their hands with this gelatin mold. And this is agar being poured into a petri dish. This is what we do for each plate that you use in the laboratory. A chemically defined media is a prepared media that has purified ingredients, and therefore we know the exact composition of the media. Therefore, we need to know exactly what the microorganism's growth requirements are in order to have good growth in this particular media. An enrichment media is a pre-prepared or equivalent media to which we have added additional growth factors. This can be a chemically defined media or others, but we have actually added things to enrich it for better growth. Complex medias are extracts or digests of yeasts, meats, or plants. What Needham actually used in his experiment was a complex media. And the problem is you don't know exactly what is in there. How much of the meat was digested by the enzyme, or how much of the plant was digested. We don't know exactly. Nutrient broth is a complex media in a liquid form, and nutrient agar is the same thing with agar added to form a solid media. This represents a chemically defined media. We know that there is 1 gram of glucose in this media. Therefore, the bioavailability, the amount available to the microbes, is 1 gram. You'll also notice that there is no agar in this recipe, and so this would be a liquid media. This is a complex media. We know that we put in 15 grams of pancreatic digestive casein, but how much of that 15 grams is actually bioavailable? We're not quite sure. This is a blood agar to which sterile blood has also been added, and you'll notice that this also has agar in it. And so this would be a blood agar plate. Culture media can be defined in several ways. Selective media suppresses the growth of some microorganisms while allowing the growth of others. Therefore, they select for certain microbes. Some media can only grow gram positives, while others only tend to grow gram negatives. So they select for one or the other. Examples of selective media include mannitol salt agar, which selects against the non-skin flora, meaning that they tend to grow skin flora. McConkey agar, which selects against gram positives, so gram negatives tend to grow. The eosin methylene blue, the EMB agar, which also selects against gram positives. And the Columbia nutrient agar, or CNA, which selects against gram negatives. So CNA tends to grow gram positives, while McConkey and EMB tend to grow gram negatives. This is an example of a selective media. Here you can see Staphylococcus epidermidis, Proteus mirabilis, and Enterococcus faecalis. Proteus mirabilis and Enterococcus faecalis are both gram negatives, while Staphylococcus epidermidis is a gram positive. So this media selected for the gram positive and against gram negatives. Differential media allows the growth of more than one microorganism of interest, but with morphologically distinguishable colonies or changes to the media. Examples of differential media include mannitol salt agar, McConkey agar, EMB agar. And go, wait, weren't those selective medias? And the answer is yes. They can be both. Blood agar is a differential media because many times different bacteria will hemolyze the blood within the agar, and this allows a differentiation. So we can distinguish or differentiate between different bacteria. For example, this blood agar plate here. The original color is here in B. This is Staphylococcus epidermidis, and this is Staphylococcus aureus. The colonies may be slightly different from each other, but they're both white colonies. The difference is Staph aureus hemolyzes blood agar, changing the agar's color to yellow, while Staph epidermidis does not. Here are actually 3 different bacteria on a plate, and they all look different from each other. Different colors, different sizes, and the presence of hemolysis. Can a media be both selective and differential? And yes, they can be. This is a nutrient agar plate. E. coli, a gram-negative, and Staphylococcus aureus, a gram-positive, both grow on this plate. Nutrient agar is also an enrichment media. MacConkey agar tend to grow gram-negative bacteria. So if we inoculate E. coli and Staphylococcus aureus on this MacConkey agar, only the gram-negative E. coli tends to grow. This shows E. coli and Salmonella enterica, which is also a gram-negative, but you'll notice that there is a difference in the color of colonies, and Salmonella tends to cause the red MacConkey agar to turn yellow. Anaerobic culture methods are used to grow anaerobic bacteria. There are a number of reducing medias that contain chemicals such as thioglycolate or oxirase that combine with any oxygen in the media to deplete the oxygen in the culture media. Typically, when these are heated, it drives the oxygen off. We use thioglycolate to grow the Clostridium bacteria that are anaerobic bacteria. Oxirase, I love the name, literally is an enzyme that erases oxygen. We can also use chambers that are anaerobic with catalysts that will drive off the oxygen. Capnophiles are microbes that require high CO2 conditions, and we can use CO2 packets or a candle jar. The candle jar, we put the inoculated media into this jar, put a candle in, light the candle, seal the jar. What happens to the candle? And how does this create an anaerobic condition? This shows an anaerobic jar, which is similar to the anaerobic pouch we use in the laboratory. In this, we have a CO2 generator. We have a catalyst that has palladium pellets that will combine with the oxygen to form water, and usually an indicator that shows us that the oxygen is gone. We use the pouches. When I seal the pouch and break the CO2 generator, this releases CO2, increasing the amount of CO2, releases hydrogen gas that combines with any oxygen in the package and forms the water. Enrichment cultures encourages the growth of a desired microbe. So again, it's important that I know the metabolic needs of the bacteria I'm trying to grow. Assume a soil sample contains a few phenol-degrading bacteria and thousands of others. We inoculate the phenol-containing culture media with the soil and let it incubate for 24 to 48 hours. We then will transfer 1 ml to another flask of the phenol media and incubate that. Do it again and again and again, and eventually only the phenol metabolizing bacteria would be growing in the media. The phenol will kill those that cannot use the phenol and will only allow for the growth of the phenol metabolizing bacteria. A pure culture contains only one species or strain of a microorganism. A colony is a population of cells arising from either a single cell or spore or from a group of attached cells, such as any staphylo. It's very hard to separate out a single grape off the cluster, but all the bacteria within the cluster will be the same microorganisms. So we tend to say a colony is often formed by a colony-forming unit, either that single cell or spore or the group of identically attached cells. The streak plate method is used to isolate pure cultures or pure colonies. We do this by taking our inoculum, using the loop, and streaking along 1/4 to 1/3 of a plate. We then re-sterilize the inoculating loop, drag it through the first streak line, and make a second streak line. We then re-sterilize. We then drag through the second streak line to make the third streak line, and if we have room, we'll actually do it a fourth time. This will allow us to dilute, if you will, the number of bacteria within each streak line, and you wind up with single isolated colonies. You know, if you take this colony and put it into other media, that you have only one type of bacteria or one strain of bacteria. We use both the inoculating loop and the inoculating needle. Do not call the inoculating needle the stabby thingy or any other type of name. It is the inoculating needle. We inoculate auger plates, deep auger tubes, and auger slants, and broth tubes. To do a slant tube, we use a method called stab the butt and streak the slant. We take the inoculating needle, immerse it into the inoculum, or take a colony from a plate. We stab down to the bottom of the tube, and then as we come back out, we streak along the slant. A broth tube will be inoculated using the inoculating loop. We take a colony or a loop full of other inoculum media and swish it around into the liquid. How do we preserve bacterial cultures for study in the future? A couple of ways we do this. One is deep freezing. This is a process in which a pure culture is placed in a suspending liquid and quick frozen at temperatures ranging anywhere from -50 to about -95 degrees Celsius. This is not just throw it in the freezer and hope for the best. This is deep freezing in either liquid nitrogen or liquid CO2, and then storing it at extreme, very cold temperatures. Lyophilization is freeze drying. Lyophilization is where we freeze in either liquid nitrogen or CO2 or another suspending liquid, and then dehydrate the frozen chunk in a vacuum. This is done via sublimation, where we go from the solid state to the gaseous state, and all we will have left is the bacteria. The liquid goes from the frozen state to the gaseous state, leaving behind the frozen, dried bacteria. Reproduction in prokaryotic cells is done by a number of methods. One, and probably the most common in bacteria, is binary fission, where we get equal cytokinesis of the original cell. Budding also occurs in many bacteria, as well as many of the yeasts, and this is where we get an unequal cytokinesis. There are a few of the bacteria that also reproduce by what we know as kinetiospores, and this is a group of bacteria known as the actinomycetes, and fragmentation of filaments. This is binary fission. We make a copy of the single circular chromosome, and we divide equally in half. How quickly can this occur depends on the bacteria. But we can go from 1 cell to 16 cells, in this case, in about 120 minutes or 2 hours. This shows those diplobacillus, and probably most diplobacillus occur simply because there is an incomplete separation of the cell walls of the two dividing bacteria. While this is a yeast, many of the coccibacteria can also undergo this process known as budding. This is an unequal cytokinesis, but each bud will have a copy of the chromosomes. The actinomycetes bacteria tend to grow more like a mold, and they produce a reproductive spore known as a kinetiospore. That's what these little curlicue fries are, as strings of these little tiny spores. They, just like a fungal spore, will be then able to spread in the environment. Also, any bacteria that tend to grow elongated chains or the molds will fragment many times. And the fragment that is broken off, such as this piece here, will then be able to move somewhere else and start a new colony. How do we measure or how do we determine how rapidly a cell is growing? Well, we look at the generation number and the number of cells. You'll see that if I put in a single bacteria in the first generation, which is 2 to the 0, I have just simply one cell. But after 5 generations, so one cell becomes 2, becomes 4, becomes 8, becomes 16, becomes 32, the 5th generation, we now have 32 cells. If I keep doing that, if I do another 5 divisions to generation number 10, I now have 1,024. And if I double that generation number, I now have 1,048,576 individuals. That's a lot of numbers. And so what we do is we convert the raw number of cells to a log number. Now, don't panic. You do not have to do logarithmic calculations. But this makes the numbers easier to look at. So here is our actual numbers of bacteria growing up to the million plus. But if we put a log scale on those numbers, you'll notice that at 5 generations, we're at 1.51. At 10 generations, we're at 3.01. We double the number of generations, we double the log number. When we go from 10 to 20 generations, we go from 3.01 to 6.02. Doubling the number of generations, double the log scale. Here's 2 graphs showing the numerical growth and the log growth over the same period of time of a bacterium. It's much easier and have a much straighter line when you do the logarithmic growth scale. Generation time is the time required for a bacterial cell to grow and divide. It is dependent on the chemical and physical conditions. If everything is great, we should have maximum growth. So, for example, if 100 cells are growing for 5 hours and produce 1,720,320 cells, we will take the log number of cells at the end minus the log number of cells at the beginning, divided by a constant 3.01, and that gives us the number of generations during this time. We then can take 60 minutes times hours and divide by the number of generations, and that gives us a constant 21 minutes per generation. This is actually the generation time of E. coli. So every 21 minutes approximately, we get a new generation of E. coli. We can then look at a growth curve of a particular bacterium or particular population. We go through 4 different stages: the lag phase, the log phase, which is an exponential phase, the stationary phase, and the death phase, which is also exponential. We'll go through each one of these separately. The lag phase, you'll notice, has no growth. It's actually a period of time with little or no cell division at the beginning of the growth curve. The cells are not dormant, they are busy going about metabolism, but are not yet reproducing. So the lag phase is a very active period of intense metabolic activity gearing up for rapid reproduction. Once we enter the reproductive state, we then enter the log phase. Period of rapid reproduction, the generation time will reach a constant minimum, such as 21 minutes per generation. However, if nothing changes in the environment, we eventually reach a stationary phase, which you'll notice is flat. This is due to the fact that the growth rate slows to a point where the number of new cells equals the number of dead cells, and the population stabilizes. This stationary phase is probably due to the exhaustion of nutrients and the accumulation of wastes, along with changes in pH. Remember, many bacteria produce acids. If, again, nothing changes in the environment, we then enter the death phase, and this is a logarithmic decline. The number of deaths now exceeds the number of new cells being produced, and this will continue until the population is diminished to a very tiny fraction of cells in the previous phase, or the culture dies out entirely. That's why when I start a culture in the laboratory for stock bacteria, I have to periodically take a few of those cells and put them into a new environment where there's plenty of nutrients and no waste products. Otherwise, the culture will completely die out. So, this shows these phases of growth. You can apply this to any population, be it microorganisms or macroorganisms. Measuring microbial reproduction, we do so in two different ways, either directly or indirectly. Direct methods will include serial dilution and viable plate counts, membrane filtration, the most probable number, the microscopic counts, which is the most accurate. Nice thing is, though, now we actually have electronic counters that do these microscopic counts for us. Plate counts, we assume that each live bacterium grows and divides to produce a single colony. However, a colony often arises from that short segment of chain of bacteria or cluster, so this is reported as a colony-forming unit. How many colonies are there on this plate? Do you want to count them? I don't. So what we have to do is get a plate that we can actually have a count. We do this by serial dilution. This is used when too many colonies grow on a plate and cannot be counted easily. By convention, we normally want a plate that has anywhere from 25 to 300 colonies per plate. I don't really want to count a 300 colony plate, but somewhere in between those numbers. To do a serial dilution, we take 1 milliliter of a sample with, in this case, a known number of microbes, mix it with 9 milliliters of sterile saline or sterile media, and continue to do this until the number of colonies that grow are within that 25 to 300 degree range on a plate. Let's look at how we do it. Here we take 1 milliliter of the original culture, put it in with 9 milliliters of broth. This is a 1 to 10 dilution. We then take a milliliter of that dilution, put it in another 9 milliliters of broth, and we have a 1 to 10 of the 1 to 10 dilution. That means we have a 1 to 100 dilution. And we continue to do this up to usually about 1 to 100,000 times dilution. We then will inoculate plates for each of the dilutions and look at the plates to see where we want to count. So, for example, in the 1 to 10 dilution, I don't even see any single colonies except for a little one right off to the left-hand side. So this is too numerous to count, TNTC. The 1/100, I see a few more individual colonies, but still can't count. Even in the 1 to 1,000 times dilution, I still see clumps of colonies that I cannot separate from each other. However, in the 1 to 10,000 dilution, I now see individual colonies, and I think I've counted these, and there's about 30. In the 1 to 100,000, however, I only see 4 colonies. Yeah, easy to count, but the problem is when I do the mathematics from the dilutions, I will not get a very accurate number. That's why we want to have 25 to 300 colony-forming units to be able to count on the plate. A pour plate is a dilution colony that is poured into the plate and then warmed nutrient agar, just ready to solidify, is poured on top, and the plate is agitated to mix the culture and the agar. This will distribute the bacteria throughout the media plate, and colonies will grow within the media as well as on top. A spread plate is a dilution colony that is poured on the top of a pre-poured solidified agar and then spread over the agar surface, distributing the bacteria equally using a sterilized metal or glass rod. The colonies only grow on top of the media. This is the difference. The pour plate, we pour the inoculum in first, add the agar, swirl it all around. The spread plate, the media is already solidified. We put the inoculum on the top. We spread it around equally with a sterile rod, and they grow on the top of the media. This shows one of these spread plates. Another direct measurement of microbial growth is filtration. The sample is filtered through a membrane that is fine enough to not allow the bacteria through. The filter then is transferred to a petri dish containing a pad soaked in the liquid nutrient media, where the colonies then arise from the bacteria that grow on the filter surface. To allow the sample to be filtered through this very, very fine filter, we usually will put the whole system under a vacuum. This will draw the liquid through the filter and trap the bacteria, such as these Staphylococcus here on this filter. We then take that filter, put it on the slightly warmed media, allow the media to soak into the filter paper, and then the colonies will grow on the filter paper. The most probable number method is based upon the fact that the greater, and that should be GREATR, the number of bacteria, the more dilutions are needed to reduce the density to the point where no bacteria are left to grow in the tubes of a series dilution. So we do an undiluted, a 1 to 10, and a 1 to 100 dilution. We will then inoculate 5 tubes from each of the dilutions and let them grow. In the undiluted sample, we had 4 tubes that grew, 1 did not. In the 1 to 10, we had only 2 tubes, and in the 1 to 100, we only had 1 tube. We then will go to a statistical table and look at the combination of positive tubes. So a 4-to-1 will tell us that approximately 26 bacteria per 100 milliliter of solution was what we had to begin with. Now you'll notice that there's a 95% confidence limit here of 9.8 to 70, meaning that there could be anywhere from 10 bacteria per 100 ml to 70. That's a pretty wide range. However, 95% of all those samples that have a 4-to-1 combination will have 26 bacteria per 100 ml. So we can say with 95% confidence that we have 26. If we had a 5-3-1 combination, we would have 110 bacteria per 100 ml. Direct microscopic count is the best way to get an actual count of the bacteria. Nice thing is we do have electronic counters that now do this for us. But when I was growing up, we actually had to do this looking through the microscope. A measured volume of bacterial suspension is placed within a defined area on a microscope slide. The bacteria are then counted in several different fields and averaged. We use a specially designed slide called the Petroff-Hausser cell or cytometer to do direct microscopic counts. Here's what this glass slide looks like. It has an area that has a 1 millimeter by 1 millimeter square, and then it is divided into 25 sections. What you will do under oil immersion is count the number of bacteria within each one of those 25 squares, add them together, and divide by 25. That gives you then an average number per the volume that was found in each of the 25 squares. And doing mathematical calculations, you can then figure out approximately the number of bacteria. So for example, my average was 14. The volume of area counted was 8 times 10 to the minus 7th. That gave me 17.5 million bacteria per milliliter in my original sample. Now, does it really make that much difference if it's 17.5 million or 17,503,489 bacteria? Not when you're talking those large numbers. So this actually is a fair representation of the number of microbes. Indirect methods of measuring microbial growth include metabolic activity, dry weight, and turbidity. Turbidity uses a spectrophotometer or colorimeter to measure the cloudiness or turbidity of a specimen. As bacteria multiply in a liquid medium, the medium becomes more and more turbid. Less light will pass through the sample, and this change will be registered as a percentage of transmission, or absorbency, optical density. This is a logarithmic expression of the percentage of transmission. I use this actually when I look to see have the bacteria grown in a stock culture. After 24 or 48 hours, I can look at the bottle and see, is there turbidity? If there is, there's been bacterial growth and reproduction. Do I have any idea how many bacteria are in that stock culture? Not a clue. All I know is that there has been growth and reproduction. In this picture, where is the most turbid solution? This is the old spectrophotometer, and we will not use these, but to do this, we would actually have to use this machine and look to see how much light got through a sample. For example, in this clear solution, 100% of the light goes through the solution. So we get 100% transmission or 0% absorbency. However, in this sample where we have turbidity due to the bacterial growth, only a small amount of the light gets through the sample and only about 20% of the light is transmitted. This will then be equivalent to 0.7 absorbency on the logarithmic scale. Metabolic activity assumes that the amount of a certain metabolic product, such as a gas or an acid, is in direct proportion to the number of bacteria present. So we can measure the amount of acid or CO2 or other gas and say, based upon how much we produce of that acid or gas, this will then correspond to approximately this number of bacteria. Dry weight is used for the filamentous bacteria such as the actinomycetes and molds. The specimen is removed from the growth media, filtered to remove any extraneous material, dried in a desiccator in a pre-weighed bowl, and then reweighed. And we simply subtract the weight of the empty bowl from the one with the dried material, and that's the weight of the bacteria or mold. From this, again, we can do some statistical analysis and determine approximate numbers. So, direct methods, plate counts, filtration, the most probable number, and direct microscopic counts either visually or electronically. Indirect methods, turbidity, metabolic activity, and dry weight. We can also today use genetic methods. This will isolate DNA sequences of many of the unculturable prokaryotic cells. Based upon our knowledge of how much DNA this particular microorganism has, we isolate out the DNA from a group of bacteria or microbes. We then can actually do statistical analysis, and depending upon the amount of DNA, we can estimate the number of microbes. This has become very helpful in those bacteria and other microbes that we cannot culture on artificial media. So, you give them food, you give them a good place to live, and what happens? This is what happens. Speak, speak. That was Bella. Thanks.